Enzyme kinetics describe how reaction rates are measured. We can use enzyme kinetics to determine how long it takes an enzyme to convert a substrate into a product. Let's look at the conversion of hydrogen peroxide to water and molecular oxygen. Hydrogen peroxide is a strong oxidizer. It's used to clean scrapes and cuts. A bottle stored in a medicine cabinet lasts for a year or longer. Inorganic catalysts, such as iodide ions, speed up the reaction rate by about 5 orders of magnitude. Let's do an experiment to see how the catalyst works. Add some hydrogen peroxide and a little soap to a graduated cylinder by dragging them to the cylinder. Add the catalyst potassium iodide. Drag it to the graduated cylinder. See all the bubbles? They result from the oxygen produced in the reaction. Ever notice how hydrogen peroxide also bubbles when you pour it on a cut? A catalyst must be present. The enzyme catalase is released when cells are damaged. Catalase catalyzes the decomposition of hydrogen peroxide to water and oxygen. Catalase has 4 subunits, just like hemoglobin, and an iron-containing heme group. It also requires the coenzyme NADP. Catalase speeds up the decomposition of hydrogen peroxide by 15 orders of magnitude. Catalase can degrade 10 million molecules of hydrogen peroxide in 1 second. The reaction rate depends on how often an enzyme and substrate collide. The catalase reaction is limited by how fast the reacting molecules can diffuse and collide with the enzyme. Reaction rates depend on the reaction conditions. Since a reaction can't occur any faster than the time it takes for 2 molecules to collide, reaction rates are sensitive to concentration. The rate of an enzyme-catalyzed reaction will increase with concentration until the enzyme is saturated with substrate. When the enzyme is saturated, all active sites are filled with substrate. In general, only initial rates are used in kinetic calculations. The initial rate can be measured in this region of the left graph, which shows change in substrate concentration as a function of time. This is important because the substrate concentration decreases as the reaction proceeds, thus affecting the reaction rates whenever the enzyme is not saturated. Temperature also affects reaction rates. In general, the rate of a reaction increases as temperature increases. Recall the combustion of sugar. However, the reaction rates of enzyme-catalyzed reactions won't keep increasing forever. At high temperatures, proteins unfold and denature. Because an enzyme's catalytic ability depends on its structure and being able to form lots of substrate-binding interactions, the unfolded enzyme doesn't work very well. Many of the reactions that enzymes catalyze involve the transfer of protons. Proton transfer reactions are very sensitive to pH. Recall that pH is a function of proton concentration in the solution. A protein's structure is sensitive to pH. Extremes in pH cause proteins to unfold. Each enzyme has its own unique temperature and pH profile. Because of the charges on substrates and enzymes, ion concentrations influence reaction rates. The ions shield the charges to prevent proteins from aggregating and precipitating out of solution. In addition to environmental conditions, molecules called activators and inhibitors affect enzyme activity. Activators and inhibitors are molecules that bind to enzymes. Activators and inhibitors are like the volume controls on a stereo. Activators turn up the volume and improve the catalytic ability of enzymes. Inhibitors turn the volume and the enzyme's activity down or off. Some activators and inhibitors change the enzyme's shape. An enzyme can have conformations with different activities. Recall how hexokinase's shape changed upon glucose binding. Let's look at a dimeric enzyme that has 2 forms, an active form and an inactive form. When an inhibitor binds, the inactive form is favored. When an activator binds, the active form is more likely. Many metabolic pathways are regulated in this way. This is called allosteric regulation. These molecules, called effectors, bind at a site other than the active site. Another type of an inhibitor is a competitive inhibitor. Competitive inhibitors compete with the enzyme's substrate for the active site. An enzyme's activity depends on its structure. Its structure depends on temperature, pH, ion concentration, and effector concentration. Enzyme activity also depends on substrate concentration and the concentration of competitive inhibitors. Many drugs exert their therapeutic effects by acting as competitive inhibitors of target enzymes.